Welcome back. This is Endoplasmic Reticulum 2, so straight into it. At the end of Endoplasmic Reticulum 1, I mentioned how the functions of the smooth ER differ from the functions of the rough ER, and we talked a little bit about drug detoxification involving hydroxylation, which is the addition of hydroxyl groups to hydrophobic drugs to increase their solubility so that they can better or more easily and effectively be excreted from the body. And I mentioned how that was under the control of a group of family of proteins called the monooxygenases, or the cytochrome P450. So let's just take a quick look at how that happens. These cytochrome P450s, these monooxygenases, you'll often recognize them because they'll have the gene name abbreviation CYP. So like CYP2E1 would be a cytochrome P450 or a monooxygenase. And what they do is they take electrons from NADH, and in an acidic environment they use oxygen to transfer one of these oxygens to this compound here. R is just a molecule that we're going to modify, and so we take one of these oxygens here and we transfer it to this hydrogen here, and that produces this hydroxyl group, and that obviously increases the polarity of the molecule and increases its solubility in water, more likely to be excreted in urine, that kind of thing, or in material, in fluids which are containing high amounts of water. That produces NADPH. NADPH is like NADH, if you remember that, as an electron carrier from cellular respiration, and NADPH here is what you heard about when you learned about photosynthesis, and this obviously produces a water molecule as well, and so that's called hydroxylation. Is what can happen in the cells of organisms is that if you increase the concentration of a drug that you're giving to an animal, for example, or a human, and the classic example of this is injecting phenobarbital into rats and increasing the dosage. That increases the expression of the cytochrome P450, the monooxygenases, which detoxify the barbiturates like phenobarbital, and that means that you have to have more drug to get the same effect. And this is because you get a proliferation of smooth endoplasmic reticulum, which is where you find these monooxygenases, cytochrome P450s. And so this is one of the ways in which people become tolerant to certain drugs. It's because the drug's metabolism increases because the amount of smooth endoplasmic reticulum increases, and there are a lot of contraindications between certain drugs and things like alcohol, because of how alcohol interferes with liver functions via the function of the smooth endoplasmic reticulum. And there's another group of cytochrome P450s which can detoxify these polycyclic hydrocarbons. Now, the problem with that is they can become more toxic than the actual polycyclic hydrocarbons. So this is benzoapyrene. It's actually one of the most toxic compounds found in cigarette smoke, and it can be detoxified in the liver by the action of these cytochrome P450s. In some cases, the actual detoxification promotes the solubility of the drug, but the actual drug that's produced ends up being more toxic. So there's a fine balance between getting this out of the body and producing too much of it, and increasing toxic byproducts. And so these enzymes which can detoxify these polycyclic hydrocarbons are often called aryl hydrocarbon carboxylases, and there are certain behaviors. Cigarette smoking, for example, will increase the expression of those, but obviously by the time these compounds, such as benzoapyrene, which are carcinogenic, have found their way into your liver, they've probably been in your lungs and all kinds of other tissues, and have caused genetic changes which could lead to cancer eventually. Another function of the smooth endoplasmic reticulum, particularly in liver cells, is the breakdown of stored glycogen. So the smooth endoplasmic reticulum has an enzyme called glucose 6-phosphatase that's unique to the surface of smooth endoplasmic reticulum. So what that does is exactly what it says it does. Actually, it's a phosphatase. So if you remember kinases that add phosphates, phosphatases like glucose 6-phosphatase, remove phosphates from substrates. So in this case, the substrate for glucose 6-phosphatase is glucose 6-phosphate. And so when you remove a phosphate from the 6-carbon of glucose 6-phosphate, like you see in this equation here, you produce free glucose, and then that can be exported from the cell and circulated through the bloodstream to other tissues that need the glucose. And so when we think about a function of the liver being glycogen storage, one of the things that might come to your mind is how do you liberate that glycogen in the form of free glucose? And it's under the control of this enzyme, glucose 6-phosphatase, which takes the 6-phosphate off of glucose and produces glucose. And so you'll see in liver cells, you'll see a decent amount of smooth endoplasmic reticulum, a lot of glycogen granules, and if the body needs glucose, then the liver can liberate that using glucose 6 phosphatase. And you see often a close apposition of the glycogen granules to the smooth endoplasmic reticulum, and so there again there's a structure function relationship. You're putting things close together that need to get close together so you can rapidly liberate glucose if you need to. So just to kind of look at that in a diagram, when glucose is needed by the body, by tissues and cells in the body, glycogen up here, which remember is a polymer of glucose, is broken down, and the released molecule is actually a molecule called glucose 1-phosphate. All the 1 means there is that the phosphate's attached to the 1 carbon of glucose. So chemistry taught you about carbon numbering in molecules. So glucose 1-phosphate has a phosphate attached to the 1 carbon, and then an enzyme shifts the 1 carbon phosphate to the 6 carbon position on glucose, and then this enzyme on the surface of the smooth ER, glucose 6-phosphatase, can remove that phosphate from the 6 position, and then the glucose can be exported out of the cell and into the blood, thereby increasing blood glucose concentrations, and cells that need that glucose can take it up using other mechanisms. So another example for the function of endoplasmic reticulum is calcium storage, and a good example of this is calcium storage in muscle cells. So muscle cells have a special kind of endoplasmic reticulum called sarcoplasmic reticulum, and so that's a good example of smooth ER that specializes in calcium storage. And so calcium is pumped from the cytoplasm into the lumen of the ER, the smooth ER, the sarcoplasmic reticulum, by calcium-dependent ATPases. And once inside that lumen of the ER, the calcium is bound to specific calcium-binding proteins, so it cannot exit again, and when it's needed for muscle contraction, because a lot of muscle contraction is under the control of calcium release, when you need to contract muscles, then the calcium can be released from the binding proteins in the ER lumen, and it will diffuse out of the endoplasmic reticulum, and that is through specific channels, calcium channels, that will open in response to certain signals for muscle contraction. Another function that we could talk about, let's think about that. We talked about cholesterol and steroid synthesis, so that's another function. And so, like we said, if you've got a tissue that contains cells that's doing a lot of steroid synthesis, so a lot of exocrine glands, for example, they would be very high, very rich in smooth endoplasmic reticulum. And in plants, we find smooth ER in plastids of some plants that are involved in phytohormone synthesis, so like the phytoestrogens and that kind of thing that you find in soy. So there's a few more functions. So let's just look at cholesterol synthesis and look at a kind of an example of a drug that interferes with that. So if you look at molecules like cholesterol and cortisol, which is an important regulatory hormone that you'll learn about if you take physiology and if you take cell signaling or endocrinology, then any other steroid hormones, testosterone, progesterone, estrogen, all those kinds of things, they've all got this kind of four ring structure. So these are carbon rings, and cortisol, cholesterol, steroids, they all have this common structure, and they only really differ in the number and arrangement of the side chains and this hydroxyl here, for example. So this enzyme, or this compound here, cholesterol, is under the control of an enzyme in the ER called hydroxymethyl gluteryl-CoA reductase. That's a big long name, don't worry about it. HMG-CoA reductase. And so that's an enzyme which is involved in reducing hydroxymethyl gluteryl-CoA, HMG. And so that's the kind of the most important step in the production of cholesterol. And so is what HMG-CoA reductase does is it takes HMG-CoA, which is just a molecule, you don't need to worry about the structure of it, and it converts it to mevalonic acid. And mevalonic acid, or mevalonate, is the critical precursor for cholesterol metabolism, this molecule on the left here. And so lovastatin is in the family of drugs which interfere with cholesterol biosynthesis. So is what these drugs generally do. This one in particular targets this enzyme HMG-CoA reductase. So it's a competitive inhibitor for HMG-CoA. And so if you give cells lovastatin, is what happens is that HMG-CoA reductase cannot bind HMG-CoA, and so it cannot make mevalonic acid, and so you end up basically stopping the synthesis of the precursors needed for cholesterol, and that lowers circulating cholesterol levels. And so that's one way in which you can lower cholesterol in the body. And then there are dietary ways and exercise ways and things as well. But that's a nice example of how a specific drug interferes with specific endoplasmic reticulum function that's biomedically relevant. Another function for the smooth ER is in eukaryotic cells, the ER is the primary source of membrane lipid kind of metabolism and regulation in terms of where the lipids go to in the cell. There are a few exceptions to that. For example, mitochondria are responsible for producing phosphatidylethanolamine, and chloroplasts generally contain the enzymes for chloroplast-specific lipids. So when I say that the smooth endoplasmic reticulum is important in membrane biogenesis, as always in biology, remember there are lots of exceptions to some of these rules, and that's just because of the way that evolution works in biological systems. So the fatty acids that you need for membrane phospholipids, they're synthesized in the cytoplasm, and they're incorporated into the ER membrane on the cytosolic side. So they're integrated on this cytosolic side of the membrane. So the fatty acids are produced, and then the phospholipid synthesis occurs, and the integration of the lipids, the phospholipids, occurs only on the cytosolic half of the bilayer. Then lipids which need to be in the other leaflet of the bilayer get flipped, and this is where membrane asymmetry is generated. So you generate asymmetry here on the surface of the endoplasmic reticulum, and that asymmetry is then transferred to any organelle which receives membrane from the endoplasmic reticulum. So the Golgi, for example, or certain vesicles, the plasma membrane, they all receive pieces of membrane through the endomembrane system from the endoplasmic reticulum. So the asymmetry you generate on the leaflets in the ER ends up being the asymmetry you see in the Golgi, and the asymmetry you ultimately see in the plasma membrane and in vesicles. Now there are some exceptions to that. Chloroplasts, peroxisomes, mitochondria, they obviously don't receive lipid directly from the endoplasmic reticulum, and so in that case they have specific phospholipid exchange proteins which help generate lipids and phospholipids in the membranes of chloroplast, mitochondria, and those kinds of things. So that's a lot more about endoplasmic reticulum, and next time I believe we'll be getting onto the Golgi apparatus. I'll see you soon.